just listen about science, but to engage with it, question it, and grow from it together. Before we begin, I would like to acknowledge the efforts of IRSA, the All India Research Scholars Organization. One of the IRSA's key aim is to create meaningful academic platforms where researchers, scholars, and students can exchange ideas, discuss emerging scientific questions, and build collaborative networks. Through such scientific talks, IRSA seeks to promote interdisciplinary sciences, expose young researchers like us to edge cutting research, and encourage critical scientific thinking. These initiatives not only support individual academic growth, but also contribute to strengthening India's research ecosystem by fostering innovation, collaboration, and a vibrant scientific community. Now, coming here to today's exciting themes, extracellular control of brain plasticity. Brain plasticity is central to how we learn, adapt, and function. Yet, many of the extracellular mechanisms shaping neural circuit remains underexplored. Today's talk promises insight into how extracellular matrix remodeling influences neural circuit networks and how these processes relate to the early dysfunction in neurological conditions, an area with profound implications for neurosciences and translational research. I am now, it is my great pleasure to now introduce our speaker, Dr. Jeet Bahadur Singh. Dr. Singh is a research associate scientist at the Center of Cellular and Molecular Therapeutics, Children's Hospital of Philadelphia, specializing in pediatric neurodegenerative and metabolic diseases, with a doctoral from the Leberg's Institute of Neurobiology, Medberg. From his research spans synaptic plasticity, retinal microcircuit networks, and inborn metabolism. He has conducted postdoctoral work in the USA and Norway and contributed to high impact publications in EMBO reports, scientific reports, and Journal of Neurosciences. His work focuses on uncovering molecular mechanism, underlying brain development and disease, supported by extensive international training and collaboration. Dr. Singh, thank you for joining for us. And the virtual floor is all yours, sir, now.
Hi, everybody can hear me?
Yes.
Hi, everybody. Good morning from here, and it's good evening there, I hope. So before I start, I wanted to mention, I did PhD from Lebanese Institute for Neurobiology. It was named after a mathematician. So just to add it on. Yeah, thanks for AIRSA for hosting us and providing me the opportunity to share what I have learned through my journey in science. And I will also thank Dr. Lal for providing this platform to all the students to have this communication going on both the ways. So with this, I would like to maybe share my screen and share. Do you see the presentation?
Yes, sir.
Yes, sir.
You can see the presentation? Okay. I think it's going a bit slow. Yeah. So my topic is like how metabolic and ECM remodeling failures derail hippocampal function. And I would like to talk about new mechanistic insights from Seelenthrin disease as a model system and how these derailment lead to structural plasticity. And what you're looking here is my setup, which I've used for my recent studies, which have been published recently. And to go from here, this is a very important slide. This is from our research group from last year. So you can see this small baby here. When he was like 2 days old, everything was fine. And then they thought that the baby was very lethargic and not very active. And then the doctors taking care of him, they became worried about it. They did some blood work. They found there was some urea disorder in his blood work. And then the doctor's team decided, and it was a kind of an emergency situation because it was life-threatening situation for this baby. And so they talked about it, and there were only two things they could do. Either he could wait for liver transplant, which was a long queue, and they generally don't give it to very young kids. He has to grow bigger enough to survive the surgery. So he had the second option, which was like nobody was thinking about it. And at that point, Dr. Kiran Musru, he's a professor and MD PhD at UPenn. He collaborated with CHOP, my research leader, Dr. Rebecca Ahrens Nicholas, and they came up with a plan to help the baby. And the plan was to do a CRISPR and target the specific gene, which was the reason for causing the whatever they were seeing wrong with his blood work and urea cycle disorder. So they thought like they could wait for 6 to 8 months while he grows big and he is still waiting in the queue to get liver transplant, or they could do something about it. And Dr. Kiran Musru's lab was already working on something on 6-7 different diseases along with collaboration with our group. So they could figure it out the right gene and the right time, and then they could do the CRISPR-Cas gene editing, and with that they could deliver the drug with the lower dose, and then they kept on increasing and with time. And now KJ is more than 1.5 years. He's surviving. He is walking. His parents could take him after like a 7 to 10 months of hospital time to back to home. And this drug was specifically designed for KJ and genetic variants in it has, and it's called personalized medicine. This is like something for the first time of this kind of thing has been done to save a patient at a very short time. And so this paves the way for future development for such ailments of rare metabolism disorders and stuff like that. And this has prepared the NIH with a framework where they can quickly approve this kind of procedure approval and everything. So this set up that kind of system. So which is like, this is why we work in science. So mostly that was delivered here from our group last year. So this was something which I wanted to share with you all before, like a motivational thing, that whatever science we do, it can come really to the patient at the site and can help them. So yeah, we should keep going on. So from here, I would like to share 2 of my papers from last year, which span into different fields, but they come together at one point. So first paper talks about CLN3 disease, which is like a biomarker for Batten disease, also known as, and it's a metabolism disorder, and which disrupts very early postnatal hippocampal maturation. And second study talks about activity-dependent extracellular proteolytic cascade that cleaves the ECM component Brevigan to promote structural plasticity. These are like the 2 of the studies which I will talk about it because these are my most recent publication and like the studies which have been completed. So to go from here, I will talk about hippocampal trisynaptic circuit. So when we talk about hippocampus, it's an organ which is in our brain, and it's responsible for most of the memories formation. And so hippocampal trisynaptic circuit has been very well studied. And in our case, we will be monitoring it by VSDI. VSDI is a voltage sensitive dye imaging, where you can record with a very fast camera the changes happening in real time in the hippocampus. But in the earlier days, around 2015, when this paper came out from Germany, from Stephen's lab, they have to be really sure where this and know the circuit, how it goes, where they stimulate. And these were something which, like, the person who was going to follow up with the experiments has to be very deep into the circuits of the system and where they are making a cut to derail the circuit and then do the treatment and everything. So this is how it was. But then what we did, and in our group, what has contributed was, like, we use now a VSDI. VSDI stands for voltage sensitive dye imaging. So I will use this term a lot. So please consider VSDI as a short from voltage sensitive dye imaging. Just remember it. You will hear it many times. So using VSDI, transmembrane potential can be recorded with ease. Spatial resolution is only the limit of the pixels in the VSDI camera, and it helps in targeting dysfunctional neural circuit. It can broadly record transmembrane spatiotemporal activity. And to show you the trisynaptic circuit in detail, because if you look at this cartoon, you will see in many of my slides. So I will like to spend some time on this one. So when I say perforant pathway, so I will be stimulating the dentate gyrus of the hippocampus, and then the circuit goes through the, what you can see through the cartoon. And when I will say like cephalocollateral, the stimulation will be happening here, and then the circuit goes on, and then we will see the output in the form. So to give you an example where I'm doing a perforant stimulation, into the hippocampal slices in the dentate gyrus region of the hippocampus. You can see, so the stimulation will happen, and you see this was 1, and then second stimulation 2, and then again we wait, there will be the third one, and then the fourth one, and this makes the circuit pass on here. And this is, of course, I have slowed down the speed so that I can show it to you guys. And so this is like the experiment which I do. And then most of the times when I will say this is like the rasters. The rasters are the drawings, geometric drawings, which we draw here along the pathways. Different pathways we talk about, like we will draw about the cephalocollateral pathway, then dentate gyrate pathway, and then the output comes in forms of the rasters. And when the rasters comes, I will show you what I'm saying rasters. And rasters are the output recording of the voltage differentiation which happens here upon the electrical stimulation. So to go from here to the next slide. So there have been some studies already done about CLN3 diseases in the past from our group. And where they have found that this Baton disease has been known to be a accumulation disorder and neurodegenerative disorder. And in order to address that question, when they looked at 2 months, 6, 7 months, and 18 to 21-old mice, this is like normal progression of the how it looks. And what I was talking about are these are the rasters, which are like the output of what of the electrical stimulation and how the camera records it. And we do MATLAB, and in the MATLAB, the output comes like this when we make the rasters. So this is like the first stimulation, second stimulation, third stimulation, and the fourth stimulation. And then it spans over the whole DG internal blade and external blade. And then this is the 6 to 7 months old mice. And then if you go on to 18 to 21 months old, this is how the circuit looks like in normal wild type mice. When there is a disease mice, in that case, you can see the activation of DG in perforant pathway stimulation leads to significant decrease in CLN3 mice already at the age of 2 months old. And then you can see that decline is persistent with time till 18 to 21 months. And this blue thing shows like high significance here, which I think you guys can see here this bar. It's like representation of the blue plot is here, which says like this is highly significant, the differences between the two groups. So this was the perforant pathway. And when we talk about the cephalocollateral pathway, which is like this pathway, when you stimulate it at this point of the hippocampus, you can see at the age of 2, 6, 7 months, 18 to 21 months, how the normal wild type mice reacts to that electrical stimulation and how the circuit behaves. But when we go to 2 months old mice in disease state, there is like hyperexcitable state here in the cephalocollateral pathway, and which is also significant with respect to comparison to the wild type group. And this thing you can follow up from 2 months to 18 to 21 months. So why this is of interest to me, because this was a background study before I joined the group here at with Rebecca. Because the loss of CLN3 also disrupts the glycerophospholipid catabolism in lysosomes, suggesting that the potential use of glycerophosphoinositol as a disease biomarker. And they show that CLN3 is required for lysosomal clearance of GPDs and reveal Baton disease neurodegenerative LSD with defect in glycerophospholipid metabolism. And this was recently the paper from 2022. And then the other group who published in 2025, they also suggested that the CLN3 loss leads to the significant accumulation of glycerophosphodiesterase at the end of the product of glycerophospholipid catabolism in lysosome. So with last 2 slides or last 3 slides, what I wanted to bring is like in the field, it has been known when we use Baton disease as a neurodevelopmental disorder or something like this, they say that it's an accumulation disorder. And the accumulation, what they have found and the studies which have been done is like they start from 2 months. So where there is accumulation and disease is already in progression. And this loss leads to, and with these 2 newer studies from 2025 and 2022 points out that there is a role of glycerophosphodiesterase and glycerophosphoinositol in making the disease biomarker. And how so if you can look the healthy and the disease state, the lysosomes looks like this, and when there is a dysfunctional CLN3 disease model, then you can see how the lysosomes are behaving. So to address this, So what we looked for, the first thing was like looking at the CLN3 disease mice at P11 age for the glycerophosphoinositol and glycerophosphodiesterase, and you can see the differences in the three between the CLN3 knockout mice, CLN3 heterozygous mice, and CLN3 homozygous mice. So you can see the clear differences. So glycerophosphoinositol accumulate in the mice, but not the glycerophosphodiesterate at the age of P11 and so this could be a GPI, could be a metabolic signature. From this data we could come to this point. And from here I wanted to address this point that there are people in the field, and the data and all the knowledge we have points towards that it's a degradation model. But here what we found out, it's not a degradation model, but it's a developmental model. And how we can target that, we did the similar study. We did the voltage sensitive dye imaging. We stimulated here for perforant pathway, and we followed. This is for the first time, of course, the previous study, that it has been shown how the neuronal network develops at P7, P14, P21. So you can see how the neuronal development happens at P7, P14, and P21 age of the mice. So you see, of course, at P7, the gating is not so done in the mice. It's a very young mice. Development is happening. So you see the signal is leaking from internal DZ to HeLS CA3. But then it somehow, around P14, it gets matured. And at P21, you can see as wild-type mice around 2 to 3 months old, I showed in the previous slides. So you can see that that's how the normal development of neural circuit happens in the hippocampus. But if there is a disease state, for here, we took the model of Batten disease, CLN3 knockout mice. So you can see at P7, you can already see there is the hippocampus of the diseased mice is hypoexcitable, means it's less excitable here in response to the perforant pathway stimulation. And at P14, there is some homeostasis happening, so it sorts out. But then you can at P21 again see the similar hypoexcitable response happening. And as I told you before, this purple thing marks the significance. And of course, you can see clear significance between the wild type and knockout mice. And yeah, so this points out that the CLN3 disease hippocampus is significantly hypoexcitable in response to perforant pathway stimulation. From here, when we go for cephalocollateral pathway, you can see using the same model at P7, P14, P21 stimulation. This is how the normal hippocampus cephalocollateral stimulation and the network looks like. And that when it's a disease model, you can see it becomes hyper excitable. So whatever we were seeing at 2-3 months is already happening at P7, P21. So it's not a neuro accumulation disorder or a neurodegeneration model, but I would say this CLN3 disease is a neurodevelopmental model from these data, that it's already starting at P7. So there's something which is happening already at the age of P7 and P14. So this signifies that it's a neurodevelopmental disorder, not a neurodegradation disorder, which is like common known knowledge in the field. So it challenges that. And from there, we also did some EEG to confirm in network dysfunction in vivo experiment model, where we implanted electrode in the P30 to P30 old mice, and then we recorded for like 2 to 4 days, and then you can see the difference delta, theta, and alpha waves. And what we found that in P30 CLN3 mice, disease hippocampus shows background slowing with an increased delta and alpha ratio, which is like marker for which you can see in several of the diseased model of neurodegeneration, stuff like that. So it's point out that there is a slowing in there. So in vivo confirmation of network dysfunction is also present at P30 age. So this marks that CLN3 disease is a neurodevelopmental disorder, and it begins in the first postnatal week for now, what we know from my data. And metabolic plus electrophysiological defects precedes storage pathology, and neonatal therapeutic window should be of target to look for the disease pathology, so where we can target it. So at the moment, there are ongoing experiments where we are using AAV mediated gene therapy, where we are injecting mice at different age to correct this disorder, and the work is still in progress. And from here, I want to jump on the next study, which we published last year was plasticity requires molecular remodeling too. So synaptic potentiation upon stimulation leads to spine formation. And how this happens, so proprotein convertases get activated. They go and activate the proteases, which go and lactican as a marker for ECM. They go and make it degrade so that new synaptic spines can work there. So, and this all requires strong neuronal activity, and no new spines, no ECM remodeling if there is a blocked extracellular proteolytic cascade. So this cartoon depicts more or less what we will talk about in the future, in the finding. And when I say ECM, ECM stands for extracellular matrix, and here lacticans are one of the family members of the ECM family, and these are present in between the synaptic junctions, and they act as a very tight sieve. And once they are present, there are no synaptic remodeling is possible. The only way to present the window of opportunity is to provide strong neuronal activity. Like, for example, people learn how to drive, how to learn to swim, even though they have grown up and the extracellular matrix have formed. So when there is strong neuronal activity, there is still the window of opportunity, and people learn over the period of their lifetime various activities. So it's not just when they are young, which is much easier to learn things than, because of the ECM, it's a very restrictive nature. It doesn't allow it. But with strong neural activity, it can be, this paper points out how we can bring that plasticity to happen and the molecular mechanisms involved in it. So with the next slide, we will like to focus on that. So what we did here was, like, this is a Western blot example representation of this graph. So you can see there, like after like 15 minutes of stimulation with picrotoxin, phospholane, and roliparam, this is like a strong stimulant, which leads to, in this case, a degradation. So like so there are like two bands which have been shown here at 145 and 53 kD. These are like the example of a brevican protein. Brevican, this is the full-length brevican protein, and this is the cleaved brevican protein. And this is, we had another antibody which specifically targets this cleaved fragment. So this is called neoepitope. So both of these antibodies have been represented here in orange and in green, and they both follow after 15 minutes of stimulation. There's increase in full length as well as degraded proteins level, and this lasts for 90 minutes, and then it comes back to falls to normal level. And to figure it out, as I said, like we need the protein. We needed the proteases to be activated, and the proteases are always present there, but they need proprotein kinases to activate. So we did plethora of these proprotein convertases, inhibitors, furin inhibitors, and PFR. And what we found, when we use them, the degradation of matrix falls down. And we also did, we could also see, like when we put a plethora of these blockers, which blocks the ADAMTS4, which is an example which we found as the target, which cleaves the brevican protein, here also decreases with time. So this suggests that the activity activates ECM cleaving proteolytic cascades, which follows like we need strong activity, and then proprotein converters have to be activated to activate the proteases, which will go and cleave these full-length proteins, and which will create a window of opportunity for the new protein, for the full-length brevican synthesis as well as the degradation of the brevican. And this degradation will give us the space and opportunity for the new synaptic contacts to happen, or whatever we have learned new to be saved. And then to go from there, we also showed that the ECM cleavage is required for the spine remodeling, but not for LTP. So on the right side, of course, this is a bit fuzzy, taken from the paper. So here we show like the LTP is not required, but because you can see TIM3, which is the blocker for the proteases, still you can see there is a LTP happening, which is the example here is phospho-ARK, and this is phosphocam kinase. So you can see there is phosphorylation happening with phosphocam kinase and phosphorylation of ARK. And here with electrophysiological, there is no significant difference between the two. Black and red are the in the presence of TIM3 is the red one, and still you can see there is a potentiation happening. So it doesn't affect the potentiation, but ECM cleavage is required for the ECM remodeling. So what we did here, these are YFP slick weak mice, and what you can see in these mice that upon they have only few neurons in the CA1 region of the hippocampus. So upon stimulation, you can see there is a synaptic remodeling happening in like you can see in control compared to PFR. There is, like you can see, long philopodias happening here. But when we put the TIM3, which is a blocker for the ADAMTS4 protein, as I said in the last slide, ADAMTS4 protein is the protein which cleaves the extracellular matrix protein, which provides the opportunity here. So in this case, when we use the blocker, there is nothing happening. It's same more or less like control. But when we use the PFR, the stimulant, in that case, you can see window of opportunity is happening at the same point. So what I wanted to say is that the plasticity requires ECM remodeling, and activity needs permissive ECM environment, and Brevikine cleavage equals to molecular switch for structural plasticity in this case. So bringing both the studies together, what we found is like development and plasticity share a common principle, which is like early metabolic bottleneck leads to the failed circuit formation, and in adult case, the ECM bottleneck leads to the failed structural remodeling. So hippocampal function depends on timely molecular remodeling, and both these studies redefine when and how intervention should target hippocampal dysfunction. So, and I would like to thank my whole group, so which have helped in the study. So Sangeeta has helped me in the EG experiments, and Devin helped me with the glycerophosphodiesterase studies, and these are all the members from my current lab. And I would like to take the question. Thanks for giving me the time to present this work.
Thank you so much, Dr. Singh, for this insightful and thought-provoking talk. It was truly fascinating to learn how these mechanisms and their broader implication for neurosciences and translational research specifically. Now we'll move towards the question and answer session, and I'll encourage all the participants to actively participate. And you may post your questions in the chat box, or you can unmute yourself and ask the questions. I'll request everybody to keep your questions brief so that we can take as much as questions possible. And with your permission, sir, we'll start taking questions from the audience.
Yeah, sure.
So we have a question in the chat box that learning slows down during the advanced age. Is it due to ECM deposits in the brain?
No, ECM keeps developing. So ECM already, so there is a stage when the ECM reach maturation. So such studies have been done in mice for long. And so, yeah, ECM deposits restricts the new synaptic function. But if there is a strong stimulation there, that's what my study shows, that it will open the window of opportunity, and new synaptic contacts and new information can be stored at that stage. I hope I answered the question.
Do we have more questions? Otherwise, sir, I have a question regarding this, but you might feel that it is a very deviated question. However, since my interest is also ECM regulation, and I'm just in the second year of PhD, and I'm facing some problem related to, as in, I'm not able to find a perfect housekeeping gene, since the gene of interest, which is in my focus area, which is bone tumors in children. My focus of gene is actually making a bond with beta actin. So I'm facing how I should select the housekeeping genes, particularly which with respect to the area that is ECM regulation and all. If you can help me with it, or what are your comments on that? I have seen that you have used beta tubulin for that, and when I have put that real time, it wasn't really working in my case. So
yeah, sorry, you have to still add to that question, or I should answer?
No, sir, it's okay.
So what I was saying, like, yeah, of course, I could not encompass all the data in the slides, but the place where you saw beta actin, that was to show the study regarding the ARC phosphorylation or the CAM kinase phosphorylation. So those were for those part. So I have not talked in detail how the ECM extraction works, So the ECM extraction, so there is like a continuous ABC enzymes which goes and cleaves the ECM that was used to isolate the ECM. So basically in the ECM, you should not have any of these contaminants when you have isolated it. So it's like a different phase, and you isolate the ECM first in that specific solution where the pH and everything is maintained in presence of continuous ABC, which you incubate the slices for like 30 minutes at around 30 to 37 degrees temperature, and then you collect the ECM, and then rest of the phase is like you do a brief spin and take the supernatant, and that contains the ECM. Rest of whatever is left is from the internal contamination. So if there is a beta actin and all these kind of housekeeping genes are present, this means it's like an internal coming from the internal components, not from the extracellular part. So I never use that. So what I used to do is like there is a paper which I can show it later, not from me, but where they what they do is like they add it's called TTE or something, like they add to the gel, and before you run the gel, it gives you the total protein concentration in each lane, and with that you can normalize the ECM level in each case. So in each lane. So like in my case, at the start of the study, I used to normalize it with the total protein, and then I was thinking there's nothing happening, as you can see in my first plot, because there was both were changing. So I couldn't even use that to normalize. It took like 1 year to figure out that part. So in this case, I would say like beta actin, tubulin, these can be used only if you are using the internal component. But if you are talking about extracellular matrix, then it's outside. So it's not there. Okay. So it's from contamination from the internal food. So this was for the phosphorylation part, not for the ECM part.
Thank you, sir. So we have one more question in the chat box. So is this possible? There is hemiplegia caused due to electrophysiology, means neuro and metabolic BMI.
I have to look at it, but I can look at it, but I don't have any comments upon that one.
Any more questions or any discussion that the panel wants to ask? I don't think so, sir. We have more questions, but is there any comment or anything from Lal sir or Vipin sir?
Yeah. Thank you so much, Dr.Jeet Singh, and you have considered our request for this such kind of wonderful discussion or talk, and really it was very informative, and so thank you so much. This is my things. So Sujata, I think we don't have the two more questions. So
yes, sir. Sure, sir. As we now come towards the end of this session, I'd like to propose a sincere vote of thanks to sir. Thank you so much, Dr. Jeet Bahadur Singh, sir, for an exceptionally insightful and engaging talk. Thank you for sharing your valuable research, perspective, and time with us. And it was truly being an inspiring session for everybody present. I would like to also thank AIRSA, All India Research Scholar Association, for continuously creating platforms that promote scientific exchange, collaboration, and professional growth within the research community. Initiatives like these play a vital role in strengthening our academic ecosystem. I would also like to thank all the participants for your active involvement, thoughtful questions, and enthusiastic presence. Thank you all once again for joining us today, and we really hope that we can plan and such more scientific discussions ahead also, and have a wonderful day, everybody.
yeah, Sujata, one more thing.
Yes,
Dr. Jeet Singh also invited in our scientific conference like the AIRSA Recon. And so somehow we couldn't connect due to some internet issue. Even you know very well that Sujata and Shrestha and our many colleagues was the managing all these things, such a wonderful conference. Really, it was good. So I appreciate to everyone and all the team members and our colleague or most welcome to our participant. You are showing the enthusiasm for such kind of scientific event, and we are conducting the every month for such a kind of lecture talk. So thankful to everyone. And Sujata, thank you so much for
Welcome, sir.
such a support, and our team member also. Thank you so much.
Thank you. Thank you, sir. Thank you, Jeet, sir.
Thank you
Thank
for
you so
providing
much
this platform
Jeet
for
Singh,
engaging this discussion.
for such kind of wonderful discussion and
Yeah.
talk. Thank you so much.
Thank you,
Thanks
sir.
for your, thank you.